Good day, I'm Dr. T. Welcome to my office. Today I'd like to talk about epigenetics. Epigenetics is kind of a top layer that goes over your normal genetic code. So we're all used to A's, T's, G's, and C's, or kind of the Mendelian genetics. Well, with that, you've got the material, you've got your promoter regions, transcription factors, everything like that. But there's actually another layer going on to control the DNA. This is called epigenetics, and this is modifying the DNA in such a way that the transcription factors either have an easier time getting to it, what we call euchromatin, or a harder time getting to it, what we call heterochromatin. And the way you do this is by controlling how tightly it's packed. So your DNA is not flopping around free. It's wound around histones, forming nucleosomes, and that those are then packaged more tightly within your cell's nucleus. Now, depending on what part of the cell cycle your cell's nucleus is, does depend on just how tightly it is packaged in there. But we're not talking about the points where it's actually packaged tightly for replication. We're talking about once it's a little bit more open and is actively making proteins in abundance. Now, in this case, we could leave it fairly well packaged up, what we call heterochromatin. And this is affected by modifying the nucleosomes themselves, so not the DNA, but the proteins surrounding the DNA, or by adding methyl groups to the C's on our DNA strand, thereby indicating that the DNA needs to be packaged a little bit tighter. Alternatively, this could be euchromatin, which is a little bit looser. No methyl groups on the DNA itself, and the histones will be modified differently. They could have things like acetate groups or acetylation taking place. Additionally, they could have methyl groups, just depending on where those are located, depends on which way it goes, phosphate groups, and other various things you could do to the histones, as well as other affiliating proteins to your DNA. Now, this has some actually really interesting applications for us. For starters, chemicals that are capable of putting methyl groups on, what are called methylating agents, these could affect one's epigenetics, and we do know that one's epigenetics does change over the course of several years. And certain things like smoking will modify one's epigenetics. This could end up creating a gene that's important for preventing cancerous cells from growing, being transitioned into heterochromatin, and therefore it is not expressed, and thus leading to potential cancerous situations. Additionally, not just smoking, but pollution and even things like stress can result in the changing of which genes are more tightly packed and which genes are more readily available for transcription. There is actually an interesting case study in the case of the calico cat. So calico cats, at least the ones that are kind of the healthy calico cats, are all female. There is a scenario where a male cat could be calico, but he is in a very, very big world of hurt because his genes are having problems. For cats, their coat color is encoded on the X chromosome. Now, for mammals, the X chromosome is a rather interesting chromosome. It's fairly large and has a lot of genes on it, but for half of that particular mammal's population, specifically the males, they only have one copy. So the promoters on X chromosomes need to be set such that enough enzymes and enough proteins that are encoded on the X chromosome are produced from one X chromosome, as would be for the entire need of the cell. Whereas most of your chromosomes, they produce roughly half of that amount of protein as you need. It does depend on what's going on, though. So that is a general trend, not a hard, fast rule. But this leads a problem for the females of the particular species. While the males have enough protein, now the females have way too much. So what female mammals generally do very on in embryo development is convert one of their X chromosomes into heterochromatin, and while they copy it and keep it on hand, they don't actually use it. Well, the calico cat does this, but the cells switch, effectively at random, which X chromosome they're going to turn off, in the calico cat's case, at a certain point. And then the daughter cells, from those cells that have been replicated, once the one X has been epigenetically disabled, will have the effects of the later X's. So that means if I have a cat cell and it decides to have, say, two alleles, or it has two alleles, a black and an orange allele, and it turns off the black allele, it'll be an orange cell, or give rise to orange coat colors. It's not actually an orange cell. Now, If, in the case of a calico cat, what happens is that some of the cells choose one, some of the cells choose the other. That way you've got some cells that, if they are producing hair, will produce black hair, whereas others will produce white hair. Or not white hair, that's a completely different gene, orange hair. The end result is that parts of the cat will be black and the other parts orange. If this happens fairly early on in embryonic development, there'll be large portions of the cat that'll be one color versus the other. If it waits a little while longer, so that the more cells have divided to start with, it'll be more of a speckled pattern where smaller splotches will be black versus orange. All thanks to which strand of your X chromosome has been shuffled off to the side using epigenetics. So with that said, epigenetics is one of those interesting things that we've really only known were a big deal for about the last 20 or 30 years, and it's definitely one that keeps cropping up every now and then. And have a wonderful day.